My name is Kelsey Abreu, and I'm going to teach about the HIV life cycle. The first thing to know about the HIV life cycle is that it has 6 steps: binding and fusion, reverse transcription, integration, transcription, assembly, and budding. This is the HIV virus, and inside the capsid, this is the capsid outside, is the RNA. These things out here are called the glycoprotein. These help the HIV bind to the white blood cell. The first step in the HIV life cycle is to bind and fuse with the CD4 T lymphocyte. The T lymphocyte is just a type of white blood cell that fights non-friendly cells. These are the CD4 receptors. Alright. The next step is the HIV enzymes convert the RNA to DNA. These enzymes are called reverse transcriptase. Here I'll draw the DNA in blue little helix things. And I'll also use a purple arrow to indicate the next step. Alright. The next step is in the HIV life cycle. It's called integration. Here the HIV DNA integrates with the host DNA. So as you can see, there's the regular DNA, and here's the HIV DNA integrated with it. It is able to do this by an enzyme called integrase. When infused with the DNA host, the integrated HIV DNA is called a provirus. And so this entire thing is called a provirus. Here it may remain inactive for a long time. Next, when the provirus is ready, the provirus uses an enzyme called RNA polymerase to create mRNA. Here I'll create mRNA. Here we'll make it blue. That's what we've been using. Here the mRNA is used to create proteins and the RNA genome, which starts to create the capsid. So here we'll make the capsid for the virus. So from here, proteins are made. It makes this really long protein chain. Whatever. This is called a protein chain. So for the next step, an enzyme called protease makes the long chains of proteins to smaller proteins, so it cuts them up, which then comes together with the RNA in the capsid to create a new virus. So that's step number 5, assembly. So here we'll make the capsid with the RNA, and all the proteins are inside of it. Lastly, whoa, sorry about that. Lastly is the budding step. The capsid will use, the virus will use the envelope from the host cell to create its own capsule. So it'll steal this and get the glycoproteins from it. And then the cycle starts all over again. And the reason it steals the envelope, the outer envelope from the host, is because it contains glycoproteins, which is necessary to bind, like I said before. Thank you.